Yes, good evening to one and all. So, here till yesterday's class we have dealt with the first part of plant growth and development, where we have looked into the actual definition of growth and how the plant growth will be, beta? Indeterminate and indefinite growth, okay, that is open form of growth because of secondary meristems. And we have clearly looked into the phases of growth, okay. What are they? Meristematic phase, elongation phase and maturation phase. And then we have looked into growth rates, okay, arithmetic growth rate and geometric growth rate. After that we have looked into quantitative comparison of growth rates, that is absolute growth rate and relative growth rates. Okay. After that we have also finished off what? Differentiation, de-differentiation and redifferentiation concepts. Okay. Now here development. Development is nothing but the sequence of events or the sequence of changes that happen in a plant from seed germination to senescence. Okay. Take down. What is development, beta? Sequence of events from seed germination to senescence. Okay. Sequence of events from seed germination to senescence in a plant life is said to be development. This diagram is given in your NCERT, which is very important, ma. So, first if you see the meristematic cell, what type of growth occurs in the plant body? Abdul, you say beta? Arithmetic growth, right, wherein one cell divides into two, of which one is meristematic and the other is permanent. Again this will divide into two, one is meristematic and the other is permanent. As such the growth happens. So, few of them are returning back to the cell division phase only, yes or no, and this growth which is happening, that is increase in cell size or increase in cell number, is said as plasmatic growth. At the same time few cells are differentiating. What are the differentiated regions that you see in the plant? Yesterday we have dealt, no, differentiated regions. What are they? Epidermis, general cortex region and endodermis, pericycle, vascular bundles, that is primary xylem and primary phloem and pith. All these are what? Differentiated regions. What are the differentiated tissues? Simple permanent tissues and complex permanent tissues. Simple permanent tissues include parenchyma, collenchyma and sclerenchyma. Complex permanent tissues include xylem and phloem. Okay. So, this is about differentiation, where here you will come across two zones. What are they, ma? Expansion, that is elongation, and maturation. In elongation phase what we have learnt? What are the changes that are happening inside the cell? Increase in cell size, new cell wall depositions and increased vascularization. And here during maturation what are the changes that are happening? Clear structural differentiation, physiological differentiation and functional differentiation as well. Now here mature cell. This mature cell will undergo what? Senescence, that is cell death. Clear, right? Understood everything? Now, so development will be on different patterns. A plant undergoing different stages of life, okay, they will respond differently to different environmental conditions and phases of growth. Okay. Same plant will show different structures, okay, responding to the environment or phases of life. That is called as what? Plasticity. Okay. Plasticity is nothing but plants show different responses take down parallelly in developmental patterns. Plants show different responses in developmental patterns with respect to either phases of growth phases of growth or environmental conditions. Phases of growth or environmental conditions. Okay. So, one example, best example of plasticity you can consider as what? Heterophily. Heterophily is one of the example of plasticity. Okay, wherein hetero means different, phily means leaf structures. Different leaf structures are shown by cotton, coriander and larkspur based on the phases of life. Okay, in juvenile phase, have you ever observed coriander? Went to kitchen any time in your home? There have you observed coriander, wherein in the juvenile phase this will be the structure and in the adult phase this will be the structure. Okay. So, heterophily is the concept wherein different leaf structures are observed on the same plant, one in the juvenile phase and one in the adult phase. Okay. So, this
juvenile one goes structurally changes itself.
Yes, it is dependent on the phases of growth. Okay. Examples include cotton, coriander and larkspur. Next is, this is observed in the aquatic plants like buttercup. It's a type of plant, I will show you later. Okay. So, here buttercup, this you can say it as environmental heterophily or habitual heterophily. Habitual heterophily. Wherein different leaf structures are observed, okay, by the same plant when it is growing in the aquatic environment and the one which is growing in the terrestrial environment. Different leaf structures by the same plant is shown based on the habitat in which it is living. Okay. Example is buttercup, and here the examples include cotton, coriander and larkspur. Clear right now? Next. Yeah. Here if you see, growth is dependent on many factors, you know that, internal or intrinsic factors, external or extrinsic factors. Internal factors include, they are intracellular like genetic factors, and they also include intercellular factors like chemicals such as plant growth regulators. And extrinsic factors you can see like light, temperature, water, oxygen and nutrition. These are the extrinsic or external factors that affect the growth. Now, we will look into plant growth regulators. I will give you 2 minutes, copy, quick. Done, right? Now, plant growth regulators. How can you describe plant growth regulators as? You can take down later also, right? Yeah. How can you describe plant growth regulators as? These are small soluble organic molecules. Okay. These are small soluble organic molecules which are produced in any part of the plant body. Okay. They show effects on the plant in their growth and development. Okay, it can be either promoting or inhibiting. Fine. So, what are plant growth regulators, ma? These are small organic chemical substances that shows effect in plant growth and development. Okay. So, this plant growth regulators can be of two types. It can be plant growth promoting or plant growth inhibiting. Plant growth promoters or plant growth inhibitors. Plant growth promoters include 3 hormones beta. Plant growth promoters means what activities they promote? They will aid in cell division, cell enlargement, trophic movements, okay, fruiting, flowering. These are the activities promoted by these hormones. Okay, what are the activities? What activities comes under plant growth promoting activities? Include cell division, cell enlargement, trophic movements, fruiting, flowering. Aging and senescence comes under plant growth inhibition activities. Okay?
Inhibition of senescence.
Yeah, inhibition of senescence is nothing but cell division, right? Huh. Now plant growth promoters. So what are these plant growth promoters beta? Auxin, good, auxin, gibberellins and cytokinins. Auxins, gibberellins and cytokinins. Listen, I'll tell you. Now plant growth inhibitors. Ethylene does both the activities, promoting and inhibiting. Most of the activities of ethylene are inhibitory only. Few of them are growth promoting. I'll tell you. Have patience. Now plant growth inhibitors. Best example of plant growth inhibitors, the hormone is abscisic acid, which is popularly known as stress hormone. Okay, abscisic acid, which is known as stress hormone. This is involved both in biotic stress, abiotic stress. Plant shows its response both for biotic stress, abiotic stress by the hormone abscisic acid. Okay, and this shows the functions. Inhibitor functions are like what ma? Dormancy, promoting seed dormancy, senescence. Senescence or aging is one and the same. These are the effects, inhibitory effects, which are shown by the plant, and abscisic acid is the plant growth inhibitor. In midst of this, ethylene. Ethylene will show both plant growth promoter activity and plant growth inhibitor activity. Okay, ethylene will show both plant growth promoter and plant growth inhibitor activity. Fine. Most of its functions are inhibitory. Clear? That is direct NCERT liner. Okay, most of the ethylene activities are what? Inhibitory.
So is it an inhibitor or?
It is both an inhibitor and promoter beta. Done? Take down quickly. Done. Yeah. So now let us start learning each and every hormone. How you have to learn? What are the aspects you need to focus for each and every hormone? First is you should be clear with its discovery of each hormone. Second is what are the precursors of each hormone. Third will be derivatives of each hormone. Fourth are uses of each hormone. Fifth is applications. These are the 5 points you have to cover with each and every hormone. Even when you are revising, whenever you are learning plant growth regulators at any stage, these are the 5 points you have to make a checklist whether you are covering for each hormone or not. Okay. So first we will start with auxin beta. Okay. Auxin. Auxin discovery we will be learning first. So all the 5 hormones whichever are discovered, they are accidental only. Nothing was intentional. But the first discovery of auxin is started by Charles Darwin and Francis Darwin. Charles Darwin and Francis Darwin. Charles Darwin and Francis Darwin, they started the discovery of auxin from the seedlings, seedlings of canary grass, coleoptile of canary grass. Okay. Coleoptiles of canary grass. Canary grass belongs to what family? Poaceae family, which is a monocot, right? What is a coleoptile? Say Kunal, what is a coleoptile? At least the diagram gurtochindi, a periphery about monocot seed. Okay, this will be your endosperm. Okay, and here this is the papery cotyledon, that is scutellum, and this is the embryonal axis. Above the embryonal axis, a covering sheet, this is called as coleoptile. This sheet above this embryonal axis is coleoptile, which is covering the plumule. Okay, and at the lower side of the embryonal axis, radicle is there. The sheet which is covering the radicle is called as coleorhiza. Okay, this is coleoptile and this is coleorhiza. Fine ma, and this is embryonal axis, clear, right? So the coleoptiles of canary grass were taken, and this coleoptiles were subjected to unilateral sunlight, unilateral light. So with the exposure of sunlight, this coleoptile has shown a phototrophic bent towards the sunlight. Okay, a tropic movement is shown, that is phototrophic movement, towards the sunlight, like this. Okay, with this phototrophic movement, Charles Darwin and Francis Darwin has said that there is some chemically active substance is present, that is showing what? Phototrophic movement, a bent towards the light. But then auxin discovery is done by F.W. Wendt. F.W. Wendt, he discovered oxygen. What experiment he has conducted? He has conducted one experiment on oat seedlings. Okay, so the coleoptiles of the oat seedlings where this active substance is present, this is decapitated. Decapitation of the oat seedlings will be done, and this decapitated part is placed on the agar block. Okay, this is placed on where? Agar block. And this agar block will be absorbing, right, the chemical substances which is present in the decapitated coleoptile will be absorbed by this agar block, and on this decapitated part the agar block is placed. Agar block is placed on this decapitated part. After some period of time, this part has shown the bent response towards the unilateral light. Okay. So with this he said that the active substance is what? Auxin. And he has also isolated auxin from human urine. Auxin is being isolated from human urine, because the indole compounds are excretory products from the human body. That is why auxin can be isolated from human urine. Fine. What auxin is isolated? Indole acetic acid. Excuse me. Indole acetic acid is being isolated from human urine. Done? Next. So first thing is done, discovery is done. Now you have to know types of auxins. We have two types of auxins, beta. Types of auxins. We have two types of auxins. First is natural auxins. Second is synthetic auxins. Okay, natural auxins and synthetic auxins. Natural auxins include what? indole acetic acid, indole acetic acid or indole butyric acid. Synthetic oxins include naphthalene acetic acid, okay, 2,4-D that is 2,4-dichlorophenolic acid and 2,4,5-T, okay, 2,4,5-trichlorophenoxyacetic acid. These are the synthetic oxins. Fine ma?
2,4-dichlorophenolic
acetic acid. Okay. This is 2,4,5-trichlorophenoxyacetic acid. These are the types of oxins. Now, precursor of oxin. What is the precursor of oxin? Here in the last table, I have given you the comparative analysis on the precursors and the derivatives. Okay. So, this table you have to note down, I will give you later, but as of now you see it. Okay. Precursor of oxin is what? Tryptophan. Precursor is tryptophan. And derivatives, what are the oxin derivatives? Oxin is indole derivative, indole compounds. Okay, derivatives of oxins are indole compounds. Derivatives of oxins are indole compounds. Clear with the discovery of oxin, types of oxins, precursor of oxin and derivatives of oxin. Now, functions of oxins. Primary function of oxin is the apical dominance. What is apical dominance? See, shoot tip is there and its lateral branches will be there, right? So, auxin presence, auxin is called as rooting hormone. I will tell you, see, auxin is called as what? Rooting hormone. But most of the auxin is secreted or synthesized. Auxin is present in the shoot position, but from shoot it will reach to root and most of its activity are done in root only. Okay, auxins will help in the formation of adventitious roots in stem cutting regions. Okay, but they are synthesized where? In the shoot region. So, most of the auxins are present in the shoot, they are synthesized in the shoot apical meristem. So, because of the dominance in auxin, there will be growth of the apical bud. Apical bud will grow in a dominant way, inhibiting the growth of lateral buds. Okay, that is apical dominance. Auxin's presence in the apical bud region will dominate the growth of the apical buds and inhibit the growth of lateral buds. That is apical dominance. And second function of the auxin is what? Cell division. Okay, cell division and cell elongation. Cell elongation, to gibberellic acid is the first enzyme. After that, auxin also. Cell division and cell elongation. And next is abscission. How auxin is related to abscission? More the concentration of auxin, less will be the abscission. More the concentration of auxin, less will be the abscission. So, the auxin concentration will be more in young fruits, young leaves, shoot buds. Okay, in these regions, in young flowers, auxin concentration will be more. So, their abscission will be less. Auxin concentration will be less in senescent fruits, dried leaves. So, abscission will be more in senescent fruits and dry leaves. And auxin plays a key role in xylem differentiation. Okay, that is promoting secondary xylem, secondary phloem formation. Okay, xylem differentiation. Clear, right? Clear with the functions of auxins? Now, applications of auxins. Auxins are applied for rooting. As I told you, auxins promote rooting in the stem cutting regions. Okay, and what type of auxins are used for the rooting is naphthalene acetic acid and indole butyric acid. Supplementing this NAA and IBE to plants will promote what? Rooting. Okay. And second is flowering. Flowering is promoted by NAA, naphthalene acetic acid, and 2,4-dichlorophenoxyacetic acid. If you spray this, then you can see flowering in the plants. Fine. Next is parthenocarpy. Parthenocarpy means what, ma? Production of seedless fruits. Seedless fruits. Okay, everywhere in the market we can find seedless fruits, that is because of applications of PGRs only, plant growth regulators. And what type of auxins we use for parthenocarpy here? Indole acetic acid and indole butyric acid. Okay, and auxins will promote weed free lawns, okay, by spraying 2,4-D, that is 2,4-dichlorophenoxyacetic acid in monocotyledonous plants. Any dicotyledonous weeds will be killed. Okay, weed free lawns will be produced. Auxins promote weed free lawns. These are the functions and applications of auxins. Clear, right? Any doubts anywhere? No. We have dealt with one hormone successfully. Okay. From discovery, precursors, derivatives, types of auxins, functions and applications of auxins. Second is what, ma? Cytokinin. Cytokinin discovery. Cytokinin discovery is done by K. Skoog. What did he observe? Okay, he has observed that, listen first, K. Skoog has observed in tobacco callus, when he is growing that tobacco callus, you know callus, no? The small undifferentiated mass of cells in the tissue. In the laboratory, when you are cultivating any plant, first you will observe this small undifferentiated mass inside the test tube. That small undifferentiated mass is called as callus. So, Skoog has observed tobacco callus, okay, has shown the internodal elongation when it is supplemented with auxin. When it is supplemented with auxin, along with auxin, it is supplemented with yeast extract, coconut milk. Okay, when these are supplemented along with auxin, this has promoted the growth of what? Internodal length in the tobacco callus, and this has increased the cell proliferation, that is cell division inside the tobacco callus. This is being observed by whom? Yeah. Later it is Miller et al. Who has said that, Miller et al. Has gave us the synthetic form of cytokinin, that is kinetin. Natural form of cytokinin is zeatin. Take down. Natural form of cytokinin is zeatin and synthetic form is what? Kinetin.
How did you find out that is cytokinin?
I will tell you. Hold on. Patience. Synthetic form is isolated, that is kinetin, that he has identified it after
Spell that name.
Miller et al. Kinetin. Okay. How did he identify that? He has autoclaved herring sperm's DNA. Herring is a fish. Okay, herring sperm's DNA. Isolated kinetin from herring sperm's DNA. Clear with the discovery? First it is K. Skoog. What did he observe? From tobacco callus, this has increased its cell proliferation and internodal length. When it is supplemented with auxin, along with auxin, yeast extract and coconut milk, when it is added, this showed that increase in cell proliferation. And it is Miller et al. He is the scientist who has given us the synthetic form of cytokinin, that is kinetin. Okay. This is being identified from herring-sperm's DNA, autoclaved from herring-sperm's DNA. And then natural form of cytokinin is what? Zeatin. Clear, right? With the discovery, natural form and the artificial form or synthetic form of cytokinin. Now, derivatives and precursor of cytokinin. Precursor is adenine is the precursor and derivatives are also adenine, but this we will call it as N6-furfuryl aminopurine. Precursor is adenine only, but it is said as what? N6-furfuryl aminopurine. Okay. And these are the derivatives of what? Adenine, which is a nitrogenous base, you know, right? Next, the key function cytokinin, the name itself is saying cyto means cell, kinin means movement or division. Okay, so the key function or the major function of cytokinin is what? Division. Okay, cell division is the main function of cytokinin. As the cells are dividing and proliferating, this will delay the activity of senescence, and cytokinin is said to be shooting hormone. Auxin is a rooting hormone, cytokinin is a shooting hormone. It is synthesized in root, but it shows maximum of its functions in the shoot region. Okay, and most of the cytokinin is present in the lateral buds. Okay. So, when apical buds are decapitated, that shows profuse branching of the lateral buds. This is an application which is used in the tea plantations and hedge making. Tea plantations and hedge making, okay, wherein decapitation of the apical buds will lead to profuse branching of what? Lateral buds or lateral branches growth, which is used in what? Tea plantations and hedge making. So, that is about overcoming apical dominance. So, with respect to the apical dominance, here you have to know about two functions beta. Those are synergistic functions and antagonistic functions. First listen, synergistic and antagonistic functions. What is antagonistic function? Two phytohormones showing opposite function, single function only, they are showing opposite effects, means it is antagonistic function by two hormones. Synergistic function, two or three hormones, multiple hormones showing synergistic effect, similar effect, same effect on a function. For example, if you take cell division, both auxin and cytokinin are synergistic in function. If you take apical dominance, auxin and cytokinin are antagonistic to each other. Auxin is promoting the apical dominance, cytokinins will inhibit the apical dominance. Understood? Any doubt? Take down synergistic and antagonistic effect. Done? Synergistic and antagonistic. Next, these are the common functions we know: growth of shoot and growth of leaves. Then, cytokinins are abundantly used in tissue culture. Okay, they are abundantly used in tissue culture. How? If in tissue culture equal concentration of auxin and cytokinin are given, 1:1, equal concentration are given, so you can see the formation of callus. Okay, if auxin concentration is more and cytokinin concentration is less, this leads to the formation of root. Okay, if auxin concentration is less and cytokinin concentration is more, this leads to the formation of shoot. Okay, and cytokinins means cell division and cell proliferation. The key activity is cell proliferation. So, naturally it is delaying what? Senescence. Okay, this overcomes senescence. This is well explained by Richmond-Lang effect. Richmond-Lang Lang effect. Fine. Take down quickly, else I will send the notes. We will go for the next hormone. Okay. Next is what? Gibberellins or gibberellic acid. So here if you see, first the discovery of gibberellins. In Japan, once upon a time, there got a disease in the rice seedlings, okay, where usual growth of rice seedlings is around 2 meters height. But then because of this disease, it is being observed that the rice seedlings has profusely grown up to 20 meters of height. Okay, that is being first reported by the scientist Kurosawa. Okay, where he said that some active compounds are being released by the fungi Gibberella fujikuroi. Active compounds are released by the fungi Gibberella fujikuroi. Okay, active compounds are released by this fungus Gibberella fujikuroi, that led to the profuse growth in length of the rice seedlings. Okay. So the active substances which are released by this Gibberella fujikuroi are what? Gibberellic acids. As such, 100 compounds of gibberellic acids were identified, of which gibberellic acid 3 is widely studied. Okay. So this is the discovery of gibberellic acid. And then what are the precursors of gibberellic acids? Acetyl-CoA is the precursor of gibberellic acid. Okay. And derivatives are terpenes. Terpenes are the derivatives of gibberellic acids. So how you will identify this precursor and derivatives? What is the difference between precursor and derivative? Why are we learning precursors and derivatives of these phytohormones?
Precursor is before gibberellin, what was it? And derivatives are which can be.
Stop. So for the synthesis of these phytohormones, there is a biosynthetic pathway followed. Okay, in your biochemistry you will I will tell you, in respiration in plants and amphibolic pathway you have learned, right? Fats, proteins and carbohydrates. There you will see different intermediates. So there is a biosynthetic pathway in synthesizing anything, yes or no? As such, there is a biosynthetic pathway for the synthesis of phytohormones also. Okay. So the first molecule which is starter of this biosynthetic pathway in the synthesis of gibberellins is acetyl-CoA. This is the starter for the synthesis of gibberellic acid. When we are reading amphibolic pathway in respiration also we have learned, right? Acetyl-CoA is the raw material in the synthesis of gibberellic acid. I have told you that point. Okay. And then when this is the starter, they will be having some derivatives. Okay. At last gibberellic acid will be produced. So these derivatives are what? Terpenes. Understood the difference between precursors and derivatives? Okay, clear? Yeah. Next. So you have the discovery, precursor and derivative. Now functions. Functions of gibberellic acid includes what? Seed germination. First is what? Seed germination. How seed germination is promoted by gibberellic acid? When seed is germinating, it requires nutrients, yes or no? So the nutrients are in the form of either starchy endosperm, or in embryo in the starchy form the complex carbohydrates form, the sugars will be present at the time of seed germination. So this gibberellic acid will help in the release of amylases. Okay. This amylases will digest complex starch or complex carbohydrates into simpler carbs or into simpler sugars. Okay. So that is how gibberellic acid is promoting in what? Seed germination. Second is bolting. Okay. This is a function. Bolting is a major function and application that we are using, nothing but gibberellic acids. If we supplement gibberellic acid to some rosette plants like cabbage, beet, cauliflower. When you supplement gibberellic acids to these plants prior to flowering, what happens prior to flowering if you supplement gibberellic acids? Internodal length will be increased, yes or no? There is huge increase in the internodal length of peduncle, pedicel and complete flower, and this complete flower will post fertilization it turns to that complete fruit or vegetable, yes or no? So, prior to the flowering when gibberellic acids are sprayed in this rosette plants, that gives increase in size of what? Cabbage, beet and cauliflower. That is the major application. And next, this increases stem elongation. This is highly used in what? Sugarcane yield. This increases sugarcane yield. Fine, right? Next is what? As gibberellic acids are induced in the synthesis of amylases, they are used in brewing industries, okay? In brewing industries, they are used in the synthesis of what? Seeds, okay? Next is it enhances sugarcane yield, quicker maturity. Gibberellic acid will enhance quicker maturity in conifers, and this is delaying the ripening, right? As it is inducing cell proliferation, this will delay ripening. So, gibberellic acids are used to increase the shelf life period of fruits and vegetables, and this will increase the fruit growth. For example, let us say the usual apple size is this much. After production of gibberellic acids, after spraying gibberellic acids prior to the fruit formation, it will increase in the fruit size of the apple. Not just apple, this is used in the grape stalk also. Grape stalk will be there na beta. Prior to the flowering, the inflorescence will be turned to what? Fruit at the end. So, when that is sprayed prior to the flowering, when it is sprayed with gibberellic acid, that will increase the internodal length, which will give us huge sized grapes. As such, we are observing many hybrid varieties in the market. That is because of the plant growth regulators only. So, are you clear with 3 plant growth promoters? Okay, discoveries, precursors, okay, derivatives, functions and applications of all the 3 phytohormones. Now, here only I am telling you with respect to seed germination, 2 hormones are antagonistic to each other, gibberellic acids and abscisic acid. Abscisic acid will promote seed dormancy, it will inhibit seed germination, whereas gibberellic acid will promote seed germination and inhibit seed dormancy. Understood? With respect to seed germination, what? With respect to seed germination, gibberellic acid is antagonistic to what?
Abscisic acid.
This requires one revision, it will be easy for you only. Just one revision is required after class. Next, abscisic acid. Abscisic acid is widely known as what? Stress hormone. Stress hormone. Okay. So, the discovery of abscisic acid is again accidental. In the year 1960s, 3 different scientists from 3 different places, 3 different laboratories, they have discovered abscisic acid as what? Abscissin, dormin, okay, abscissin, dormin, and there is one more, abscissin beta, beta abscissin. Okay. So, 3 different compounds were identified, and later they got to know that all these 3 are one molecule only, that is abscisic acid, abscisic acid. Okay. Beta abscission. Okay. So, later they got to know that these 3 different active substances are nothing but one compound, that is abscisic acid. Fine. So, this is about the discovery of abscisic acid. The precursor of abscisic acid is violoxanthin. Precursor is violoxanthin. Once you look into this, yeah, abscisic acid, precursor is violoxanthin, and these are the derivatives of terpenoids. Okay, derivatives of terpenoids. Fine, clear with the discovery, precursor and derivatives, right? Now we will look into its functions and uses. So, as it is a stress hormone, anytime any stress that happens to either any organism anywhere, what is the first thing we will do? We will shut the doors, right? Same way in the plant, the first response is what? Stomatal closure. This is the first response, okay, that is observed in plant whenever there is stressful condition. Second is what? Abscission, okay, and third is, see, abscission is what? More the concentration of abscisic acid, more the concentration of abscisic acid, more will be abscission. So, with respect to abscission, again two hormones are antagonistic to each other. What are they? Auxin. More the concentration of auxin, less will be abscission. More the concentration of abscisic acid, more will be the abscission. So, these two are antagonistic to each other. Next, seed dormancy. Abscisic acid will promote seed dormancy. Whenever a seed is in unfavorable conditions, not favorable for its growth, okay, it will be in the dormant condition. So, seed dormancy is promoted by abscisic acid. With respect to seed dormancy, two hormones are antagonistic. What are they? Gibberellic acids and okay. Next, it inhibits the metabolism. Okay, stomatal closure might be, abscission might be, seed dormancy might be. It is inhibiting metabolism or promoting metabolism? Inhibiting the metabolism. That is why cell activities are stopped. Fine. Whereas metabolism will be at high in rate with the activity of cytokinin. More cytokinins are produced, more nutrient mobilization will be there. Okay. Yes. With respect to metabolism, cytokinin is antagonistic to what? ABA. More nutrient mobilization in the presence of high amount of cytokinin. Okay. Next, applications. If you want to apply gibberellic, if you want to apply abscisic acid, it will promote what? Seed dormancy, and this is an anti-transpirant. Fine. Next, ethylene, last phytohormone, that is ethylene. Okay. Ethylene is first observed as a gaseous hormone, wherein ripened oranges by Cousins, by the scientist Cousins, he has observed that ripened oranges, when placed around unripened bananas, this got ripened. Okay, there is some gaseous exchange between this ripened oranges to unripened bananas, which made the unripened bananas to ripen. Okay, that is the observation which is said by the scientist Cousins. Okay, and what is the precursor of ethylene and ethylene derivatives? Derivatives are not there because this is a gaseous hormone. And what is the precursor is? Precursor is methionine. Methionine. Clear? Any doubts anywhere? Next. Lastly, the functions and applications of ethylene. Functions and applications of ethylene. Ethylene promotes flowering and femaleness in flowers. Okay? Ethylene promotes flowering and femaleness in flowers. Example, pineapple. Fruit ripening. This promotes fruit ripening. Triple response is shown Best example is rice seedlings. Okay. Triple response is shown in the rice seedlings specially. First response is increase in the girth of the hypocotyle, okay, and formation of apical hook and swelling region, okay. Increase in growth of hypocotyle, swelling of the hypocotyle and formation of apical hook. These are the three responses shown by ethylene, especially in the rice seedlings. And then it promotes senescence and adventitious roots formation also. Applications. More ethylene is applied, it will promote fruit ripening. And synthetic form of ethylene is what, ma? Can anyone say? Synthetic form? Synthetic form of ethylene is ethaform. Ethaform is the synthetic form of ethylene. And then abscission and thinning. Ethylene promotes abscission and thinning. The more thinning of the wall layers, the more ripened it gets, right? And then increase in fruit set. As it promotes flowering and femaleness, more fruit set is increased in the plants when ethylene is sprayed, okay? And then sprouting of the storage organs. In storage organs, sprouting is observed with the spray of ethylene, fine? So, these are the functions and applications of ethylene, clear, right? So, now in our last slide, we will exercise the complete precursors and derivatives of all the phytohormones. The first phytohormone we have learnt is what, ma? Auxin. Auxin precursor is what? Tryptophan. And derivatives are indole compounds. They are indole derivatives. Second is what? Cytokinin. The natural form of cytokinin is? Artificial form is? Kinetin. Kinetin is artificial form, natural form is zeatin. And where do you observe cytokinin naturally? In corn kernels, in coconut milk, okay, yeast extract. Here you will see the natural form of cytokinin, okay. So, they are what derivatives? Adenine derivatives, which is a purine nucleotide, and they are adenine derivatives only. Precursor is adenine and they are adenine derivatives. In your NCERT there is a term given, what is that? N6 furfuryl aminopurine, okay. Next, third is what, ma? Gibberellic acids or gibberellins. Of them widely studied is what? Gibberellic acid 3. Precursor is what? Acetyl-CoA or mevalonic acid, okay. And the chemical composition or the derivatives is terpenes. Gibberellins are terpene derivatives. Next, fourth is what? Abscisic acid. Abscisic acid precursor is violoxanthin and the chemical composition is what? Sesquiterpenoids, okay. And then ethylene, okay. Ethylene precursor is methionine, which is a gaseous unsaturated hydrocarbon, C2H4, fine, right. So, with this we have finished out what? Plant growth regulators completely and development as well, fine, clear, right. With this plant growth development is done, and in our next session we will be starting with cell biology, okay. That is all for today, beta. Thank you.